Hey everyone, and welcome to the video lecture for Biology 169, Unit 1, Module 1.1.3. And this module will cover the thyroid gland and all of those associated structures and hormones. Don't forget about the lecture objectives here. Make sure that you're paying attention to these and using them to help you kind of drive your study for the lecture quizzes as well as the lecture exams. All right, let's jump into it. The thyroid gland curves across the anterior surface of the trachea, just inferior to the thyroid. It's kind of a shield-shaped gland, cartilage, which forms most of the anterior surface of the larynx. And you can see that in the figure over there to the right. The two lobes of the thyroid gland are united by a slender connection called the isthmus. And you can feel the gland with your fingers. So if you ever go and get a thyroid test at your general practitioner, they can typically feel for enlargement in the thyroid gland just by palpitating that area. So when something goes wrong, it typically becomes visible and it enlarges and distorts the surface of the neck. And the size of the gland varies depending on your heredity and environmental and nutritional factors. But its average weight is around 34 grams or so. And extensive blood supply gives the thyroid gland a deep red color. The thyroid gland actually contains large numbers of thyroid follicles, which are just hollow spheres lined by a simple cuboidal epithelium. As we start to look through all of the histological slides in the lab, you can start to see that's how the cells start to shape out. Oftentimes, we see the actual individual cells themselves surrounding a larger area, which we know as a follicle altogether, but it can kind of give the appearance of cells. So the follicle cells surround a follicle cavity that holds a viscous colloid, which is just a fluid containing large quantities of dissolved proteins. A network of capillaries surrounds each follicle, and they deliver nutrients and regulatory hormones to the glandular cells and accepting their secretory products and metabolic waste. There's a trick to identifying the structure of a thyroid follicle. If you're looking at it in a microscope, it looks similar to that of a gel capsule. The simple cuboidal epithelium is comparable to the capsule itself, and the colloid to the capsule's viscous contents. Follicle cells synthesize a globular protein called thyroglobulin and secrete it into the colloid of the thyroid follicles. The thyroglobin molecules contain the amino acid tyrosine, which is the building block of thyroid hormones, some of which you can see here. The formation of thyroid hormones, however, involves some basic steps. The first step, iodide ions are absorbed from the diet by the digestive tract, and the bloodstream delivers them to the thyroid gland. TSH-sensitive carrier proteins in the basement membrane of the follicle cells actively transport iodide ions, so I negative, into the cytoplasm. Normally, the follicle cells maintain their intracellular connections of iodide that are many times higher than those that are in the extracellular fluid. In the second step, the iodide ions diffuse to the apical surface of each follicle cell, where they lose an electron and are converted into an atom of iodine, or I neutral, or I0 if you're looking at it in chemistry, by the enzyme thyroid peroxidase. The reaction sequence, which occurs at the apical membrane surface, also attaches one or two iodine atoms to the tyrosine portions of a thyroglobulin molecule within the follicle cavity. In the third step, tyrosine molecules with attached iodine atoms become linked by covalent bonds, forming molecules of thyroid hormones that remain incorporated into thyroglobin. Thyroid peroxidase probably carries out the pairing process. The hormone thyroxine, also known as tetraiodide or T4, contains 4 iodine atoms. A related molecule called triiodide or T3, contains 3 iodine atoms. Eventually, each molecule of thyroglobin contains 4 to 8 molecules of T3 or T4 hormones, or both. If you want to see that, you can see both of those right here. These are the hormones that I was talking about previously, but I wanted to get you the steps that you see there. So the major factor that controls the rate of thyroid hormone release is the concentration of TSH in the circulating blood. TSH stimulates iodide transport into the follicle cells and stimulates the production of thyroglobin and thyroid peroxidase. The TSH also stimulates the release of thyroid hormones, and under the influence of TSH, the following steps occur. So we move into the fourth stage over here. Follicle cells remove the thyroglobin from the follicles by endocytosis. In the fifth step, lysosomal enzymes break down the thyroglobin, and the amino acids and thyroid hormones enter the cytoplasm. The amino acids are then recycled and used to synthesize more thyroglobin. In the sixth step, the released molecules of T3 and T4 diffuse across the basement membrane and enter the bloodstream. About 90% of all thyroid secretion is T4, and T3 is secreted in comparatively smaller amounts. And in the final step, about 75% of the T4 molecules and about 70% of the T3 molecules entering the bloodstream become attached to transport proteins called thyroid binding globules, so TBGs. Most of the rest of the T3 and T4 that is in circulation is attached to transthyretin, also known as thyroid binding prealbumin, or TBPA, or 2-albumin, one of the plasma's proteins. Only the relatively small quantities of thyroid hormones that remain unbound, roughly about 0.3% of the circulating T3 and about 0.03% of the circulating T4, are free to diffuse into peripheral tissues. Equilibrium exists between the amount of bound and unbound thyroid hormones. At any moment, free thyroid hormones are being bound to carriers at the same rate at which bound hormones are being released. When unbound thyroid hormones diffuse out of the bloodstream and into their tissues, the equilibrium is disturbed. The carrier proteins then release additional thyroid hormones until a new equilibrium is reached. The bound thyroid hormones make up a substantial reserve. The bloodstream normally has about more than a week's supply of thyroid hormones. TSH plays a key role in both the synthesis and release of thyroid hormones. In the absence of TSH, the thyroid follicles become inactive, and neither synthesis nor secretion occurs. TSH binds to plasma membrane receptors, and by stimulating adenylate cyclase, it activates key enzymes involved in thyroid hormone production. Let's talk about the function of thyroid hormones. So, thyroid hormones affect most every cell in the body. They enter target cells by means of an energy-dependent transport system, and inside a target cell, they bind to receptors, 1 in the cytoplasm, 2 on the surface of the mitochondria, and 3 in the nucleus. So, thyroid hormones bound to cytoplasmic receptors are essentially held in storage. If intercellular levels of thyroid hormone decline, the bound thyroid hormones are released into the cytoplasm. The ones that are in the mitochondria increase the rate of mitochondrial ATP production, and the binary receptors in the nucleus activate genes that control the synthesis of enzymes involved in energy transformation and utilization. One specific effect is the accelerated production of sodium-potassium ATPase. Recall that this is a membrane protein that ejects intercellular sodium and recovers extracellular potassium that we talked about when we covered the action potential in 168. And this exchange uses large amounts of ATP. So thyroid hormones also activate genes that code for enzymes involved in glycolysis and ATP production. Coupled with the direct effect of thyroid hormones on the mitochondria, this effect increases the metabolic rate of the cell. The effect is called the calorogenic effect, calor meaning heat, of thyroid hormones because the cell consumes more energy and generates more heat. In young children, TSH production increases in cold weather, and the calorogenic effect may help them adapt to cold climates. This response doesn't actually occur in adults. In growing children, thyroid hormones are also essential to normal development of the skeletal, muscular, and nervous systems. The thyroid gland produces large amounts of T4, but T3 is primarily responsible for the observed effects of thyroid hormones, a strong, immediate, and short-lived increase in the rate of cellular metabolism. At any moment, T3 released from the thyroid gland accounts for only 10 to 15% of the T3 in peripheral tissues. However, enzymes in the liver, kidney, and other tissues convert T4 to T3. Roughly 85 to 90% of the T3 that reaches the target cells becomes the conversion of T4 within the peripheral tissues. We obtain iodine from the food that we eat. Each day, the follicle cells in the thyroid gland absorb about 120 to 150 grams of iodine, which is the minimum dietary amount needed to maintain normal thyroid function. The iodine ions are actively transported into the thyroid follicle cells. As a result, the concentration of I inside thyroid follicle cells is generally about 30 times higher than that in the blood. If blood iodine levels rise, so do the levels inside of the follicle cell. The thyroid follicles contain the most iodide reserves in the body. TSH stimulates the active transport of iodide, and the resulting increase in the rate of iodide movement into the cytoplasm accelerates the formation of thyroid hormones. The typical diet in the United States provides approximately 500 grams of iodide per day, which is about 3 times the minimum daily requirement. Much of the excess is due to the iodine that's added into table salt, known as iodized salt, which is consumed in large quantities in processed foods. For this reason, iodide deficiency is seldom responsible for limiting the rate of thyroid hormone production. This is not necessarily the case in other countries. The kidneys remove excess iodine from the blood, and each day the liver excretes a small amount of iodide, about 20 grams or so, into the bile, which is an exocrine product stored in the gallbladder. Iodide excreted by the kidneys is eliminated in the urine, and the iodide excreted in the bile is eliminated in the feces. The loss in the bile continues even if the diet contains less than the minimum iodide requirement and can gradually deplete the iodide reserves in the thyroid. Thyroid hormone production then declines regardless of circulating levels of TSH. The thyroid also contains a second population of endocrine cells, and these cells are the C, or the clear cells, or the parafollicular cells. They lie sandwiched between the cuboidal follicle cells and their basement membrane. They are larger than the cells of the follicular epithelium and do not stain as clearly. So when you're looking at these under a microscope, they're going to be much more clear than any kind of other stained cell that you see there. C cells produce the hormone calcitonin, or CT, which helps to regulate Ca2 plus concentrations in the body fluids. We introduced the functions of that hormone back in 168, but the net effect of calcitonin release is a drop in the calcium concentrations in body fluids. Calcitonin has a few functions. One, it inhibits osteoclasts, which slows the rate of calcium release into the bone. And two, it stimulates calcium excretion by the kidneys. The control of calcitonin secretion is an example of direct endocrine regulation. Neither the hypothalamus nor the pituitary gland is involved. The C cells respond directly to an elevation in the calcium concentrations of the blood. When the concentration rises, calcitonin secretion increases. The calcium concentration then drops, eliminating the stimulus and turning off the C cells. Calcitonin is probably most important during childhood when it stimulates bone growth and mineral deposition in the skeleton. It also appears to be important in reducing the loss of bone mass, one, during prolonged starvation, and two, in the late stages of pregnancy. At that time, the maternal skeleton competes with the developing fetus for calcium ions in the diet. The role of calcitonin in healthy, non-pregnant adults is relatively unclear, though. So we've talked about overall the importance of calcium in controlling muscle cell and neuron activities. Calcium ion concentrations also affect the sodium permeabilities of excitable membranes, and at high calcium concentrations, sodium permeability decreases and the membranes become less responsive. Such problems are relatively rare, though. Problems caused by lower than normal calcium concentrations are equally as dangerous, though, and much more common. When calcium ion concentrations decline, sodium permeabilities increase, and as a result, cells become extremely excitable. If calcium levels fall too far, convulsions or muscular spasms can result. And the parathyroid glands and the parathyroid hormone are largely responsible for maintaining adequate calcium levels, and we'll talk about that soon. So, lastly, I just want to mention that thyroid hormone production is just a classic example of negative feedback loops in the body. And you can see the details outlined in that diagram there. Remember the difference between a negative and positive feedback loop while we're on the subject. Negative feedback means that there's a change in direction. If something's too high, something gets released to change the direction and bring what was too high back down, and vice versa. It's like moving to the right and then turning around and going left, or going to the left and then turning around and going back right. Students often get confused because we associate positive, negative in mathematical terms, and the confusion sets in when we consider that something in a low quantity and then the response happening, which then in turn leads to an increase because we would overall think that was positive. We went from a smaller number to a bigger number, so that should be positive, right? Well, no, because if you look in this example that I show you here, If T3 and T4 are low, the hypothalamus releases TRH, which will release TSH by the pituitary. So your gut reaction might be that this is positive feedback because we had to get a net overall gain of TSH. And that would be incorrect because we started with a low number and went higher. That's technically a change in direction. And any change in direction is negative feedback, whether it goes from negative to positive or positive to negative, or a small number to a large number, or a large number to a big number. Any change in direction is negative feedback. Positive feedback occurs when we continue moving in the same direction. So it's like stepping on the gas pedal and getting even faster. So one of the classic examples of positive feedback in the body is the release of oxytocin during childbirth, which we mentioned briefly in the previous module. So as we wrap this one up, some things I want you to keep in mind as you're studying for your quiz. What signs and symptoms would you expect to see in an individual whose diet lacks iodine? And when a person's thyroid gland is removed, signs of decreased thyroid hormone concentration do not appear until about 1 week later or so. Why?